Hello everyone and welcome back to What's Next, the world of endotoxin. I am Oya and today I'm welcoming you from a very nice city, Vienna.
Hello, everyone. I'm Brendan, and Oya, I'd have to say I'm very jealous. Vienna is one of my favourite cities. So today we're on episode 6, and what we're going to do today is look at rapid endotoxin detection methods in pharmaceuticals.
Exactly. And our guest for today's episode is Johannes Oberdorfer. He's a senior scientist for rapid microbiology methods in a pharmaceutical company. Welcome, Johannes. Would you please shortly inform us about your experience?
Sure, Oya and Brendan, and thank you to having me here. So I've been working since 15 years now in the pharmaceutical sector, and most of my time within an innovative field with automations, robotic and alternative and rapid methods. So research, development and quality control have been my position so far. And during my time as a scientist, I've been implementing now automated and alternative BET technologies, and as well as looking into alternative BET methods. So that's my experience so far.
That's great. And so, Johannes, tell us, why should we use alternative and automated methods, especially with BET technologies?
Brandon, that's a really good question, and thanks for this one. So yeah, you should ask yourself, why changing a running system or a running team? But the rate of change within our environment, well, at least in pharma, has accelerated significantly, especially when you think of the pandemic situation and the digitization and digitalization that we're facing. One thing maybe I have to clarify at the beginning is the terminology, because we are using these terms like rapid, alternative, automated methods interchangeable. And at least for the interview, because I will refer to this quite often, I would use the term with the same intention, referring to a more sensitive, more accurate, precise, reproducible test result when compared to conventional methods. So most of these techniques often do not give a fast result, even if they are called rapid, and therefore you should clarify this one. But they provide more information or other benefits. Well, and yeah, here we are already. So why you should use or better implement these rapid or alternative methods? The alternative methods usually involve some form of automation, and the methods often capture data electronically. So they offer a wide variety of advantages, including labor efficiency and error reduction. And reducing errors is one of the greatest potential benefits, at least within the pharma sector, and especially with endotoxin testing. Well, as you know, investigations, deviations, out of expectation results, they lead to a huge investment of resources, so time and money and persons you have to involve, also your interfaces. And well, this is exactly what you would like to save with your rapid method, isn't it, Brendan and Oya?
Yes.
While some methods require extra human intervention and thus create greater potential for mistakes, keep that in mind, others automate the most error-prone processes. And for the BET technologies, there are some automation solutions available, but the majority of our labs out there still rely on these labor-intense experiments. And well, I think the most of the colleagues that are listening could give us a lot of examples on this. But there are also other reasons why you should use these rapid methods, and I just give you some more. But you have the possibility to make critical business decisions more quickly. You overcome existing method-related problems, sure, and improve the test failure rates. So you reduce your retesting and all the things that come with this aspect. You reduce method variabilities, possibility of complex product matrix testing. So you can also include in-process sample testing, and the detection of impurities and also their, let's call it side effects. And sure, I mentioned already the recovery of higher or more accurate results. So talking a lot. So in a nutshell, you will need alternative and automated methods for designing or implementing a more robust process that could reduce the test failure rates and assisting with continuous process and product improvement. And well, in my opinion, that's exactly what we need in the 21st century with the continuous change and digitization ongoing, isn't it?
Yes, exactly. And you kind of summarize the main and every reason why we need those rapid methods. Exactly. And my next question would be then, what would be the strategy to implement an alternative and automated method?
Yeah, logical question here, and thanks for this one. Oh yeah. So maybe also first, I do think there's not the one specific strategy or answer. So there's no one fits all approach for the implementation or the strategy. You will have to determine for yourself your purpose, your approach, and also for your goal, what strategy would be the best. So I can just give a more on a holistic view on this strategy. And there are also regulations and guidances out there for rapid methods in the USP and the European Pharmacopoeia, and also in respective BET monographs. So the USP 1223 or the the European Pharmacopoeia chapter 5.1.6. But yeah, I do not want to bother you with the regulations and stuff. So out of my perspective, there are 3 areas where you could build your implementation strategy upon. And the first one would be, so, well, I'm from pharma, so that's for sure obvious, a cost-benefit analysis. So what do you want to achieve, and what budget do you have? What technologies are available out there? Also, what technologies are mature? And who else is using them? I know it's hard in the pharma sector to get to know other colleagues there, but yeah, it's important to do so. How rapid is the rapid method? Yeah, and especially in the times today, what papers have been published on the subject, and are these independent? Are they reliable? Are there other things that contribute to this? And what have regulators said? This is the business cost benefit analysis, and then you can go on from this on to a more detailed assessment. So to have to look at factors like time, accuracy, and automation. So time taken to prepare the test. So is it really a rapid method faster, equivalent, or slower than your conventional method? What time do you need for the test to be taking place? The sample throughput, very important. Yeah, doesn't help if you can just analyze one sample instead of 20 samples. Time to result is important. Yeah, and also the result reporting. Is it simplified, or do you have to put in more time? So the accuracy is another topic, and I think that's or should be obvious that for an alternative or rapid or automated method, your accuracy should be increased. So lesser human error, reduction in human errors, a reduction in the subjectivity. So at a real a mean value, let's say it this way. So also whether there is a need for the rapid method to detect what a conventional method cannot detect. And I think there we are struggling with the rapid methods, sometimes, or alternative methods. And maybe the last thing that I want to really point out is there a need to connect apertures and equipment with the LIMS system, so the Laboratory Information Management System. And here, just out of own experience, you should focus, okay, what is my IT department doing, and can they work with the rapid methods? So that's it. So keep in mind what your aim target picture looks like, and then you should be able to build your best fitting strategy. And that's it.
Yeah. I think that's a really good idea with regard to alternative methods. Obviously, we work in what we could say an extremely conservative area, but obviously for good purpose when we're talking about the safety of humans. And, there's one sentence that I'd say I don't despise it, something that I don't like to hear, and it's always because we've always done it that way, which, it shouldn't be the reason to continue to do something. So in regard to that, Johannes, what will the future bring to us in regards to rapid alternative methods? You can talk about sterility or BET or any the idea there. So
Thanks, Brendan. Well, yeah, I just also challenged it, thinking about the sentence. So we've always done this way. It's really the thing that we get challenged. So as a rapid methodologist, read quite often. But yeah, I think that there needs some mind change here. So, but yeah, sorry, was dragged away by this one. So no, what does the future bring for me personally? The future will be bright and shiny. So yeah, for sure, not only for the endotoxin methods, and also yeah, I think as a rapid methodologist, I'm really looking forward to see these recombinant technologies with regards to BET advancing and helping to achieve these 3 R's principle. So replace, reduce, refine, to fulfill, yeah, well, I would also say my responsibility for sustainability. And I think also most of the companies have these yeah, target picture or future perspective of being sustainable. And especially if you look like events with the pandemic, the climate changes, and then these catastrophes also happening all over the place and all over the globe. So that's our responsibility here, and I'm happy that we can look into the future, yeah, with these new technologies. And then also another topic of the European Pharmacopoeia supporting the recombinant technologies, and also having or giving the statement lately that they want to completely replace the rapid pyrogene test at this, yeah, and this is something I think, yeah, as a biological lab assistant by education, that you can drive the monocyte activation test, give it a huge boost and implement it into the pharma environment. Also, it is available in the pharmacopoeias for quite some times, but this will be, yeah, I'm really looking forward to this, that we get into an, that we can replace also this, yeah, rapid test model, which is for patient safety reasons. So it is not done, yeah, because you want to, but you have to and ensure that the patients are safe. So just for the reason. And also, what I personally look forward to is these proceeding digitization and digitalization. So I really like the point of getting automated solutions, getting the robotics. Yeah, after quite some time, where innovations and new methods weren't really developed in the microbiology environment, as you were mentioning also, Brendan, that we are more in a conventional yeah, environment here. And so I'm pretty sure my job as a scientist within a rapid microbiome team will be getting very exciting and, yeah, will be also sustainable, let's say. So yeah, I'm really looking forward to the future, and also happy to be a part and a part-taking thing of the future.
Yeah, great. So thank you so much for these informations about why we need rapid methods, why we should rapid methods, and how should be the strategy. I think in our everyday life, for people like us in a kind of healthcare industry, is really good points. And so thank you for all the viewers as well watching us. And coming next episode, we'll be running an endotoxin essay. And if you would like to go back to our previous episodes, please click on the link provided below this video. So, Brendan?
Yeah, thank you, Johannes. Before that, I just wanted to say thank you again, Johannes. I also wanted to highlight what you said about the EDQM. Hopefully, if we have any EDQM members watching, I would love for you to come on and tell us about this decision to remove the rabbit pyrogen test. It would be great to hear about it. But as we always say at the end of our meetings, please get vaccinated. Wear your mask if you need to these days. Wash your hands and stay safe. And thank you, Johannes, and thank you, Oya, for coming.
Yes, thank you.
You're welcome. Thank you also. Bye-bye.
Bye-bye.